Greetings, thank you for your interest in my lab. My name is Mick Edmonds, and my lab focuses on the role of non-coding RNAs in the initiation and progression of human malignancy. Now, we're two parts. We are both an RNA biology lab, and we're also a cancer biology lab. And really, our goal is to understand how these newly discovered genes in our genome, which isn't junk, what their functions are, how they act by themselves, how they act with other known oncogenes and tumor suppressors within the cell, and then really can we capitalize on those. And so as far as research goes, we really do have the emphasis in the lab that it has to be translational. We really start with the patient. We have hundreds and hundreds of frozen patient samples that we've collected here at UAB, Vanderbilt, Ohio State, University of Virginia, and other places, UPenn. We have great clinical collaborators, and we collect high quality tissue specimens. And we look at those. We look to see simply what genes are up, what genes are down, what mutations are common for patients with this type of disease or this specific type of pathological presentation. And then we try to understand, okay, if we manipulate it in cells, what does that do? Does that change growth? And so we start with the patient. We start with data mining. We also do data mining in our lab, mining publicly available databases, and we verify that with our own clinical patients at the bench, and students get to get a piece of human tissue and extract nucleic acid from that. And then once we develop the ideas of what genes are being changed, importantly we want to know, does that correlate to outcome? Can we look at a gene and say, if you have high levels of this, what is your overall 5-year survival for this disease? Or if we look at a gene pattern, a set of genes that are up and a set of genes that are down, can we leverage that to know within the next year or two, is this patient likely to relapse or not to relapse? With the clinical data, the cell-based molecular biology and biochemistry, and animal work, we are able to put ourselves in a high-impact publishing group. And so that's really what we look for, is high-impact papers that will set up trainees for their career. That's the type of research that we're interested in doing. And I really take a hands-on mentoring approach. I meet with all my trainees once a week individually, where we go over data, we talk about our CR issues, professional development, talk about plans for next week. It's really a way to make sure we don't lose weeks and months in a student's PhD training. Thank you for your interest. If any of this sounds appealing to you and you want to move forward and talk about a rotation, send me an email, mickedmonds@uab.edu. We'll set up a quick time to talk to each other over the phone, and then if we feel like we want to move forward, we'll set up a Zoom interview, and we'll talk about different projects available for rotations, people that you could be working with on this project or other projects. But feel free to reach out to me if you have any questions about any of this information we went over. Again, congratulations. Welcome to UAB. Go discover something and have fun during grad school.